So today we have Dr. Rice with us, and he's a professor at University of Oklahoma, and I also actually got my doctorate under Dr. Rice, so he's responsible for me. You guys all owe him a big applause and thanks for that, right? But anyways, Dr. Rice got his bachelor's and master's degree at Illinois State University. From there, he went on to get his doctorate at Purdue University under Dan Raftery's lab. And there he worked with NMR. He had two main areas. He looked at the catalytic effects of trichloro, or the photo decomposition of trichloroethane on titanium dioxide. And he also looked at the hyperpolarization with xenon as a means to enhance weak NMR signals. Since then, or after that, he went on to do a postdoc at Dr. Schaefer's lab at the University of Washington in St. Louis, at Washington University in St. Louis, where he studied NMR and bacteria cell walls and biological relevant NMR experiments. From that, he went on to become a professor at OU, where he continued on with his NMR specialties and the biology materials interface. I guess I'll turn it over to you.
Okay. Well, thanks, Jason. I appreciate the invitation to come up here and see Jason. We've been meaning to do this for a couple of years, and there's always been a conflict or something. Different schedules and get interrupted, but I'm glad I came up here in April after all the rains, because everything's nice and green and all the calves are out laying in the fields and it's pleasant. And then tonight I'll be up here for the tornadoes and the hailstorms, so this will be great. So I'm excited about it. So being from the Midwest and being from Oklahoma, I'm not scared of tornadoes. I know all about them. So it's kind of it's the second football season in Oklahoma, if you think about it. So Jason came to my lab. He was my very first graduate student. He graduated in 5 years. The experiments I'm going to tell you about today are experiments that Jason started in about 4 or 5 years ago. And we've slowly made progress on them. And so I'm here to give Jason an update and give you an update and tell you exactly what's been going on. So this work's been trickling along and kind of collecting some data, and all these people have had a role in collecting that data. But the real person who got this work going in the last 6 months is Amy. And Amy is an undergraduate researcher in my lab. And Amy is doing experiments that have never, ever been done in the history of the world, as far as we know. So it's really great to have undergraduates who can do research, and I know people here have research experiences. So don't think of yourself as just being a lab technician to go wash dishes. You can make an impact on science. So that's what I'm going to tell you about today. My training is in physical chemistry, and I learned all about bacteria, and this is my picture of a bacterium as a physical chemist, and that it's a circle. Okay? We can approximate everything with a circle. We know, the radius and so forth. And the chemistry of what makes up the cell is everything in the cytoplasm that most of microbiology is worried about. Where the DNA is, what protein synthesis, everything that's going on. We're interested in the outer membrane and the outer cell wall of gram-positive bacteria. And the cell wall is a very rigid structure. It's very heavy. It's a barrier for the cell to protect it from the outside world. So it has this network that's called peptidoglycan. And it looks a lot like a chain link fence. And it's held together with carbohydrates and amino acids. And in between are these little vines. And these vines in between the chain link fence are called teichoic acid molecules. And they're on the outside of the cell. And they have a variety of chemical functions, a variety of biology. And what we did, and what Jason helped me, was to study how this molecule, teichoic acid, helps a bacteria, for instance, stick to a surface, because this is the outside of the molecule of the bacteria, and this is like Velcro. So this is Velcro on the bacteria. It sticks to everything. It grabs everything. And so that's what we were interested in, is how it does its chemistry. So here's the structure. It's a phosphate. And it's not only one phosphate, or 2 or 3, but there are 40 or 50. So this is a huge polymer. It's like DNA. So DNA has a phosphate backbone, so phosphate, 3 carbons, phosphate, except it doesn't have nucleotides, it has sugars and amino acids. So it doesn't have a double helix, it doesn't have base pairing, it's not a stable structure, it's just a big floppy polymer molecule, which is fine because it has to weave itself in and out of this peptidoglycan framework, and so it can have that ability to twist and turn and do a lot of different things. The role that it has in biology is varied, and what we're interested in our lab is metal-ion binding, because the metals have to get from the outside solution into the cell wall, into the interior where they're used by enzymes to make proteins. And so the teichoic acid reaches out into solution and grabs onto a metal ion with the phosphate group here, and then helps bring that metal ion into the bacterium. So it has these metal binding sites, and we were interested in that. And we had done some NMR studies, nuclear magnetic resonance, and we're doing phosphorus NMR, which is very different than typical proton NMR that you would see in organic. And what we did, this is some work that Jason did. Okay? So we collected NMR data for teichoic acid molecule of magnesium, and we got a spectrum like this. And everybody in the literature from Nature from 35 years ago, even more now, thought the structure would be like this, and it turns out this structure is wrong. This actually has a magnesium through a single oxygen, and a more correct structure has it in a bidentate or chelation model, so 2 oxygens go out and grab the magnesium at once. Okay? So what we did is we reanalyzed these samples with different methodology, and we got a new answer. And so when you have new data and new theories, you have to understand, maybe you're stepping on other people's toes, maybe there's a controversy. Here's our chelation model of magnesium, 2 oxygens, and a phosphorus. And there's been other work done on RNA, for instance, where it has phosphorus, oxygen, magnesium, and this is in a structure that was done by a guy I know, Paul Carey, at Case Western Reserve. And here you can see there are actually water molecules attached to the outside. So this is magnesium with a bunch of water on it, whereas our sample does not have any water. And so what we are wondering is, because our sample is dried and these are hydrated samples, should we put our sample in water, see if we can get a bunch of water molecules in here, and do we change our chemistry? In fact, there would not be a controversy. It's just the difference between a dried sample and a liquid sample. So what we did is we wanted to dissolve our sample, and we wanted to study the water. And so we wanted to study this phosphorus, and we wanted to use a technique called Solidstate NMR, which we won't have time to go over with today, but basically it's an experiment that is described by the works. They are solid samples. So you don't have any chalk because you don't have a chalkboard in a classroom anymore. But is there a piece of chalk here? Those are pens. Yep. No, I just wanted a piece of chalk. Oh. I need a piece of chalk, but that's okay. So we'll use a piece of pen. If you put a pen in water, it does not dissolve. If you put bacteria cell walls in water, they do not dissolve. They are insoluble, just like a piece of chalk is insoluble in water. So we have to use solids because these materials that we study are solids. And so if we're going to add water and put this in suspension and try and dissolve our molecules, or at least get them hydrated, but yet we want them to be solid, what we need to do is freeze our sample. And so we need to take our liquid state sample and turn it into a block of ice. Okay? And so we're going to freeze our sample, and we're going to have a frozen solid, and it's going to be at minus 50 degrees C. And so we think that at minus 50 degrees C, the sample will be frozen. So we set this up, and so this is Jason's big job. I'm sitting in my office, I come up with these ideas, and I say, Jason, let's dissolve our sample, put it in the NMR, and freeze it. And then you go run the NMR, and we'll be famous. So Jason does the experiment, and this is, we're hoping to get, if I can go back a few slides, we're hoping to get something that looks like this. A bunch of peaks that we can understand as an NMR expert what these peaks mean. Okay? So we're looking for a signal. We dissolve our sample, and there's what Jason comes to me with. Nothing. Okay? And Jason's first thought and my first thought is he broke my very, very expensive $400,000 NMR spectrometer instrument. And this is like, okay, Jason, you're not going to graduate until we can pay this off. So. This is not a good day. This was, what are we doing here? This is in water, minus 50 degrees C. We have no signal. And I said, well, Jason, you obviously forgot to put the solid or the liquid solution in the tube, right? It's an empty tube. No, I really did it. So, well, you forgot to do something. So we sat there for a couple of days. I looked over at his shoulder. We did the experiment, and lo and behold, there was no signal because the sample, which we thought was frozen and would be solid, was in fact not solid. There was in fact liquid water in our sample at minus 50 degrees C. And why that's important is because our sample would allow for molecular motions in solution that would prevent it from being a solid. And that's the easiest explanation I can give you right now. But basically, if you're trying to do solid state NMR, you have to freeze your sample. So when we did this, basically, is this teichoic acid antifreeze agent that provides liquid water at minus 50 degrees Celsius? I mean, that was our first thought, and we were trying to figure out what can we do. So one of the things about discovering antifreeze agents and new properties is that you get distracted from your original goal of metal chelation. So that's a good thing. Serendipity is a good thing, but it's a situation where you now have more experiments to do, which always makes it exciting to be a scientist. So, what we wanted to know is, does liquid water exist at low temperatures in the presence of teichoic acid? Teichoic acid is our Velcro polymer on the outside of the cell wall, and if this has an antifreeze property, maybe that helps bacteria survive when they're frozen, and we'll get to that study. Get to those studies in proof, but as a preview of where we're going, basically we're working with NASA, and if NASA finds life on Mars and it's so cold, how can it survive? Maybe they use antifreeze molecules like teichoic acid to help life survive on Mars. Okay, and so we'll get there in our journey. So this is how we get involved, Dr. Wickham, is that we had to decide, if this is an antifreeze agent. Why is that? And so right now NASA is looking for conditions on Mars. This is the North Pole of Mars. And this is water ice. And there's also carbon dioxide ice on there at certain times of the year. And so NASA is digging down in the soil. And they have found liquid water in the soil and all sorts of salts and oxidizers and everything else. They're wondering, is there going to be life there? And so they're sending a rover right now. You've heard about these little rovers that are driving around on Mars. They're supposed to last for 3 months, and they have little solar cells on them, right? And now they've been there for, 5 or 6 years or something. So they decided, let's do this again. Let's not send a little rover. Those little rovers are the size of, would fit on this table, or 2 of them would fit on the table side by side. The rover they sent is the size of a car. Okay? It's big. It's like a Volkswagen Beetle or a Mini Cooper or something. Okay, it's not a big Cadillac Escalade, but, it's a car. It's big. And you can't use solar panels to get the energy, so it's actually nuclear powered. So it has a nuclear reactor on it. So it's pretty cool. I was out at the Jet Propulsion Laboratory in California and I've actually seen the rover. It's pretty neat. Okay. So there's maybe life on Mars, but if you actually go to an iceberg and you dig out in the middle of an iceberg, you can take this material, melt it, put it on a petri dish, and life will emerge. Microbes, yeast, bacteria will grow because there is life inside of icebergs. And so what are those mechanisms, the chemical mechanisms necessary for liquid water, and our teichoic acid is part of the answer. So here I am, a physical chemist. We're with Jason. He's a physical chemist, or an organic, or jack of all trades now. Okay. I'm studying these molecules. They have biological impact, but I don't know any biology. But I do know that they have they're antifreeze agents. So we can use our expertise as chemists to understand water. And while you may think it's very simple, and it is, there are still a lot of things yet to be learned. So, fortunately, we don't have to go to the polar regions to study frozen bacteria. We can take our bacteria, we can put them in dissolve them in water, freeze them in little ice cubes in the lab, and study them with our solid state NMR and carry out a lot of interesting studies. So, This is a picture I found on the internet. You can actually get this on a t-shirt if you want. Okay. So here we have, in nature, you can have antifreeze agents from brines and soils. In the laboratory, you can add ethylene glycol or glycerin. But in nature, it doesn't have DMSO floating around as an antifreeze agent. So what if you do if you don't have brine or soils or slime? It was also, and so survival of bacteria at low temperatures was already known. 1938, effective freezing on bacteria. That bacteria would die when they're frozen. Sometimes they live. One way you can get them to live is this. In the early days of microbiology, nearly 100 years ago now, is what they would do is they would take cultures of bacteria and they would add fat-free milk or glycerin or glucose as an antifreeze agent and help those bacteria survive freezing. And the way they would test them is they would go down to the Quincy Market Cold Storage and Warehouse in Boston, which is going down to Walmart or some other grocery store, and say, can I, go into your grocery store and put some, frozen bacteria back there? And they all freak out, Oh my God, it's next to the frozen hamburger or something. Okay. That's very different how we do science today, but it's always fun to read these old papers. So okay. What we're going to do is we're going to study our teichoic acid with deuterium NMR spectroscopy. And this methodology is very powerful and very useful. And the one person who thought of this idea is Jason. This is not my idea, this is Jason's idea. We're standing in the hallway. I can remember the conversation, and I said, what are we going to do? And Jason said, let's use deuterium NMR. And I went, wow, that's a great idea. Okay? So it was the perfect idea, the perfect analysis method. And so when we do that, we can take deuterium NMR of liquid water, but now we have deuterons in there instead of hydrogen, so now it's D2O, and this is heavy water. This is an isotope, so the deuterium has an extra neutron in the nucleus, or a neutron in addition to the proton, so now it has a proton and a neutron in the nucleus instead of just a proton and hydrogen. So it's heavier, so it's heavy water. And at 25 degrees C at room temperature, you get a nice narrow line, and you say, wow, that's nice. But what happens when you freeze it, and this is where the diagnostic properties and where Jason's insight was very useful, was that when you freeze it, this narrow line is split into this broad pattern with two peaks. Okay? And so you either have this broad double peak shape, or you have this narrow shape, and there's really nothing in between. So it's very diagnostic of the presence of liquid water or ice. Okay, so this allows us to study our molecules. So what we did is we added teichoic acid in our heavy water, froze it to minus 25 degrees Celsius, and we have our narrow liquid water peak, and we have our broad, little double signal at the bottom, our doublet, and that's the signal for ice. So even at minus 25 degrees C, we have liquid water, and this proves, at least our hypothesis that liquid water was present in the sample. Remember, before we tried to do a different NMR experiment, and we got no signal, and I said, oh my God, Jason, we broke the NMR, what's going on? And then now we know that it actually is liquid water that makes the sample have antifreeze properties. But it's not totally antifreeze. It's not completely liquid water, it's more like slush. Okay? So this is like an icy or a snow cone or something, right? So that's what we're looking at. And if you go to really cold temperatures, say minus 50 degrees C, you take this signal and it gets a lot smaller and you get more ice. So you can freeze out the teichoic acid. You can get colder and remove all the liquid water. So there is a temperature effect. So what this shows is that the teichoic acids are definitely antifreeze, or if not really antifreeze isn't the right word, they at least protect liquid water or ensure liquid water. We don't know the exact chemical mechanisms yet, but that's the future studies in our lab. But this would allow the bacterium to survive in the icebergs because it allows nutrients and waste products to go in and out of the bacterium because there's liquid water everywhere. So here's what we have. We think we have this picture of ice and water, so we have, droplets or little pockets of liquid water surrounded by ice. And so now we need to take, is this picture correct? So what we turn to is a different methodology called fluorescence microscopy, where we could actually take a picture of the ice and see what it looks like. So what we're going to do is we're going to use fluorescence to take a picture to get the size, shape, density of the water pockets. And to do that, we have to do some fluorescence spectroscopy, a fluorescence method. No longer can we rely on heavy water, but we need to add a dye. And there are fluorescent dyes all around. There are fluorescent dyes in your highlighters. Okay? So if you took this highlighter out and you dissolved it in chloroform, you get all the dye out and it turns nice and yellow, and that's really neat. Okay? So it's a fun experiment. So, the dye that we chose was rhodamin B, and this is the structure of rhodamin B as a fluorescent dye. And there's a picture of it in a test tube, a cartoon, because if you hit it with light, the light causes the sample to glow, to fluoresce, and so you get fluorescence emission from the dye. If you take the lower part of the tube and you freeze a section of it, this frozen section will no longer fluoresce, it will no longer glow, but the liquid portion on top will continue to glow. And so here's a picture that I found in a book from 1957 where they did the exact same experiment. So again, you'd think something from the 1950s, does it have any relevance to today? Of course it does. So you can take this rhodamin B, it's not glowing here, but here it's glowing, and they have a color photograph. So, and so the reason that we are seeing the color here is because this is liquid water with the rhodamin B fluorescence, whereas down here is ice and there is no fluorescence. And you can even see there's a few droplets of water that are coming down the side. Okay, so apparently they spilled a little bit in trying to do the experiment. Okay, which I thought was kind of neat to look at. So, trying to put this inside a little box, get it lined up. Oh, darn, I spilled a little bit, but that's okay. So here's our fluorescence microscope that we used actually at the Health Sciences Center. And this is a picture that's pretty boring. It's black, obviously, and there's maybe some streaks of light, but it's really hard to see. And what this is a sample that just has our rhodamin B dye only, and we've frozen it at -20, just like in that previous test tube picture, and there's really nothing glowing. Okay, I'll show you in the next picture where that really should light up. But we have a little bit of streaks in here, and those are just be concentrated regions of the dye. Yeah, that'd be great, thank you. And so if you then take this sample, and then you add our antifreeze agent, our teichoic acid, we're going to add our special molecule to this. It should provide liquid water, which then should cause this slide to light up. And that's exactly what's going to happen. There we go. So now, the actual difference in light intensity between these two factors, we had to really open up the iris and the gain on this sample. The light intensity differs by a factor of 1,000. So these are really, really bright. Okay? And we see that we have all these bright little pockets and little channels. Okay? And all the dark regions are ice crystals. Oh, that's fine.
So folk can see how to write.
Oh, of course, students have to. Okay, excellent. So what we have is we have these pockets where our special teichoic acid molecule is, along with the dye. So it's the dye that's causing it to glow, not this teichoic acid molecule, but they're together in the liquid water. In between are these ice crystals. And so you can kind of see little shapes of ice crystals, and maybe there's a few ice crystals here, and a few aligned along here, and they have different shapes and morphologies as they're all just kind of clustering together. And so it's very similar to the ice and the chunks of ice in this bag. This is not a solid chunk of ice. This is many, many different small portions of ice, and they're all lined against each other, but they maybe have different orientations and they're lying on top of each other in different ways. So this is our teichoic acid. I can put the label on that. So this is our rhodamin B with our teichoic acid at 30 degrees. And again, we have liquid water because of the fluorescence. Okay. Here's some bacteria that we put in the microscope slide, and here they are frozen, and you can see the Bacillus subtilis bacteria is a little rod-shaped bacterium. And if you look at the microscope slide and you let it warm up, if you capture images over time with a little movie, you can actually see the sample change. And what we are showing here is that the sample is actually melting. And this little piece in the upper right-hand corner is actually liquid water. And this is ice. And I wish I could get the movie to play. It's a really big file. But what we see is this gradient, this wave of liquid water going across the bacteria. And what's happening is all the bacteria in these channels here and all these channels here are actually being squeezed from the ice out into the water. So you can see these bacteria being squeezed out into the water. Okay? And here they are room temperature, all the bacteria are swimming around. Okay? So it's a really neat experiment. But let's see what happens if we're looking for life in ice. Let's add our teichoic acid molecule to this, and what we're going to see, I want you to remember this slide and how we have a few little pockets here, but these big channels here. And if we add teichoic acid, we now get a totally different morphology. We have huge ice crystals here in dark, and these little pockets of ice crystals here. I don't know what these little black nodules are, if they're like little fingers coming off the ice cubes, or micro ice cubes, or ice crystals, or I don't know exactly what's going on. It's very fascinating. And if you take this sample, and now what we're doing is we're warming it up, we're going to get pockets of liquid water, we're going to get ice around it, and you can see them begin to form and really make their little mini droplets inside of there. And so what we're thinking is that this is the sum of the properties of life in ice. So, okay, so here's a quick summary. We have our channels with our dye. We have our bacteria that's in there. I'm going to skip a couple of slides in the interest of time. One of the theories for the evolution of life is that the Earth was covered by giant ice sheets 650 million years ago because they found rock formations at the equator that could only be formed by a glacier. And how do you get a glacier down in Panama? Well, maybe it was covered with ice. Okay? And so if that's true, how did life survive and obviously reemerge? Antifreeze molecules could play a part. If there's life on Mars, they need to have antifreeze molecules. So, while not proof of life on Mars, it certainly provides information that maybe it's possible for life on Mars. So, a little bit of an update. Our model of metal chelation by teichoic acid was incorrect, which led us to study liquid water, which led us to discover antifreeze behavior, which led us to do fluorescence to show the interesting channels. But really, you know, every time I talk to people over the past few years, they'd all say, great, this is wonderful. But what does it have to do with microbiology? Maybe this is just an anomaly and it has nothing to do with life and doesn't really matter, and who cares? Because really, if this is a biological molecule that's protecting liquid water, it should protect life at low temperature. And we haven't actually done that experiment. And this is where I got in trouble because I'm a physical chemist and I'm like, I don't know. How do you test for life? You know, how do you do this? You know, get a little life meter and, you know, put it down. You know, they don't talk to me. You know, they don't send email. They don't Twitter. Okay, so I don't know what to do. You know, I can't communicate with them. So, it was easy for me to come up with chemistry studies. Percent liquid water versus concentration, a mode of action. What's the role of these branches that are either an alanine or a sugar branch? Do other polymers have the similar property? So those are easy chemistry studies. The harder studies for us were the biology. For instance, does teichoic acid help bacteria survive when being frozen? And what you need to use is a thing called a cell viability assay, a test for life. And then, if it does protect life, does it protect different species of bacteria? Does it affect other organisms? Does it help yeast survive? And can we get bacteria without teichoic acid, in which this teichoic acid is deleted in a mutant bacteria? So we've eventually worked on all these projects over the last couple of years, but it's fascinating with science is that you get to learn new things. And this is where students are wonderful, because students go out and they do the experiments and they help with the literature searches and they figure all these things out. So what we need to do is we need to find an assay that was easy to use, because we're not microbiologists. I certainly am not. Okay? It needs to be easy to understand. Now this was hard because I'm a chemist. And while I teach physical chemistry, we do some analytical chemistry. And you could find some protocols out there in the literature, and you'd go to the company and they'd say, give us $100 and we'll give you this little kit. and you mix chemical A and you mix chemical B and you shake it and you see if it's red and you put it in the fluorescence and you record a number. And I said, okay, I like that, you know, shake and bake chemistry, get a number. But what does that number mean? Okay? And so I would talk to the company and they'd say, don't worry about it, just record the number. Okay, well, let's do a control. Let's take our bacteria and let's put them in the autoclave and let's kill everything. Do our shake and bake experiment, you get 0 number, right? Well, no, we still get a 50% response rate for bacteria that are dead. And I'm like, what do you mean? How can it be dead and a positive response? They said, well, don't worry about it. I'm like, okay, no, we are not using this methodology because I don't understand it. Okay, so this is where you get in trouble when you ask too many questions. Okay, how does this work? How do I understand it? Can I make a calibration curve? So it needed to be easy to implement. So we found some other tests. For instance, ATP. Life has ATP, adenosine triphosphate. That's the energy molecule of life. You've probably seen that in your biology classes, maybe, or microbiology. At least heard of ATP, right? People have probably heard of that. So ATP is easy. They would say, we have this assay. You measure the amount of ATP. It turns one molecule, turns it into bioluminescence, and it glows, and you measure the light that comes off. And I say, great, because then we killed our bacteria, and we had no ATP generation, and therefore we had no light. And I said, beautiful, we got this is working. All right, we're on board. We have a methodology. It's easy to use, to understand, and implement. And then we added our teichoic acid polymer, and our light generation went through the roof. And I couldn't figure it out, except that teichoic acid is full of phosphate, just like ATP is. So as soon as we add our cryoprotectant, we had huge interference signals. So, back to square one. So, we never gave up. So what we found was an assay that's actually been in the literature for a very long time, uses rizazirin, and it's undergoes cell metabolism to resorphorin, resorphorin. I don't know. I'm a physical chemist, so we'll just call it R. Okay? So I like giving things abbreviations. Okay? And it's pink. Okay? So this dye has two important factors. When it's blue, originally, it undergoes cell metabolism in bacteria that are alive, and it ends up being pink. So you can see the tube change from blue to pink. And that's a quick diagnostic for your eye. It's easy to see. And the pink molecule is fluorescent, which means I can measure a signal intensity. I can put it in a fluorescence spectrophotometer, hit the button, get an intensity. And if I add twice as many cells, I get the number that's twice as big. And so I really, really like that. And so cells that don't metabolize, dead cells, nothing happens. And so it's very diagnostic for life. So our goal was to quantify viable cells. The need for metabolism requires that you incubate the cells, which is fine. And if you're interested in using this, you can use phosphate buffered saline, or we've done it in just our liquid media broth, our luria broth that we grow bacteria in, and that's actually what we freeze them in. So here's what we're going to do. We're going to take our cell culture. We're going to transfer it to a 96-well plate because we have a plate reader, because we have to make lots of measurements. We're going to add our cell titer blue. It's going to go from blue to pink after you incubate it for 30 minutes. And we're going to measure the fluorescence intensity. Excellent. So we needed to make a calibration curve. So this is a calibration curve where we took our bacterium, and then we get a response here for 100% of our bacteria, and then we dilute it by a factor of 2, the signal drops. You dilute it by a factor of 2, the signal drops again, dilute it, you get this drop in signal, and if you plot this, you get a nice linear line. So great, I can now understand this. This is a linear behavior. I can now use and understand how this is working. And if I had, no cells, I get no signal. And that was also very important. Okay. So, we're ready to use our assay to measure bacterial death after freezing. So, here we are back to the scientists in Antarctica. And over here on the left is actually the professor who's reading the user's manual of how to perform the assay. In the middle is the student who's actually recording fluorescence. And there's another student in the background who realizes they're all in trouble. So. So we're going to take our culture. Now we're going to take our culture, put it in little Eppendorf tubes, which are little plastic tubes with nice little snap caps, and we're going to freeze them. So this outer box represents a freezer. We just use a standard laboratory freezer at minus 20 degrees Celsius. We're going to take them out of the freezer after a week, thaw them out, put them on the plate, add our dye, incubate, record fluorescence. So here's what we get. We did our bacteria 1 week at minus 20 degrees Celsius. What we can do is we got a response that looked like this. Now this sample is actually a laboratory standard protocol where we add glycerol as a cryoprotectant. In every microbiology lab, when they make freezer stock, as they call it, or freezer standards, they take their bacteria and they add glycerol to antifreeze, and they get a response that looks like this. And what we've done is we've taken our bacteria out. This says incubation time at the bottom. But our bacteria, when they come out, they give a signal like this, and they're in media, they're in broth. They have lots of nutrients, and they start to grow over time. So we leave them in the incubator for up to 8 hours or even overnight, and the bacteria grow, and then they level off. And this is called stationary phase. But our bacteria are growing because they have media. So not only can they metabolize, they can grow and double and function as bacteria. And so that was important for us because even though a bacteria may come out of the freezer and it might be alive, it may not be very happy. It may not want to do anything. It may not divide. Maybe you damaged some of its DNA or something. So what we wanted to do is then repeat this, but without any cryoprotectants, without any additives. And if you do that, you get the peak on the bottom. So what this shows, if you don't add anything, bacteria die when you freeze. But they don't all die. There's still a few down here. This is a really small signal. This is in the noise. And it kind of fluctuates around in the noise, so after about 4 or 5 hours, it starts to take off. And what this means is we freeze, I don't know, 99%. Maybe it's 99.999%. Maybe it's, maybe there's 1 part per million of bacteria that survives. Okay. That 1 ppm of bacteria, 1 out of a million, will sit there and divide to go 1 to 2, 2 to 4, 4, keep doubling and doubling until after 5 or 6 hours you begin to get a sizable cell culture. And eventually over a couple of days this would ramp off and get to be a large sample size. So we can kill bacteria, but we're not killing everything. There isn't complete death. And the reason there isn't complete death is because bacteria have teichoic acid, the antifreeze agent. So this is where you're thinking, boy, you should get better survival. But when you freeze bacteria live in nature. They're with their friends. You're going to, have bacteria sitting in some pond. It's going to get cold. Pond's going to freeze. The bacteria is still going to be there. Maybe their friends die off. Okay? And maybe they leave behind the carcass and the teichoic acid and everything else. Or there's other slime and other things there. So what we can do is we can maybe see if there's some intermediate range. We could add 1% glycerol. And here the bacteria will, there's increased survival over no additives, but not as much as if you add 10, but you still get cell growth. So this is why microbiology uses 10% glycerol instead of 1, because you get, 3 times more cells. But if we add our special molecule, this is the experiment that we've been waiting for 4 years to do. I was very nervous. We had our assay, and here's what we get. We had teichoic acid, and our bacteria lived. Yay! Okay? They're not initially as good as glycerol, but they're certainly better than adding nothing. And not only that, they grow rapidly. So, if this is 1 in a million, this is down in the noise, maybe I can't even measure that. Maybe this is 0 or really close to 0. And maybe this is really 10% survival. Okay? If you want to assume this is about 100% survival, this is like 375 or so, and this is maybe 75. So, maybe that's, 10, 20% survival. I don't know. And not only that, the cells come out and they're happy. How do I know they're happy? Because they're growing. They don't come out of the freezer and they don't have frostbite. They're not just sitting there going, I don't want to do nothing. Okay? They grow. So, they grow up to a point, and then they start to die off. And they die off because they're running out of media. This is the same tube of, we're not adding extra media. They use up all the nutrients. I think the reason these don't die off so well is because they're using glycerol as a nutrient. So glycerol, bacteria can grow on glycerol, use it as a carbon source, So in here, maybe they've used up all the sugar, whereas here they're using the glycerol as a sugar and using it to grow. Okay? So this is great. So I was very happy. Amy was like, is this good? I'm like, this is great. So she couldn't believe it. So I said, great, let's go do it again. And she looked at me like, I don't want to do it again. I have good data and you're happy. I don't want to do it again because it may not work again. Well, it did work again. Okay? We did it again and we got nearly identical curves. There's a little bit of variability because it's just a different bacterial stock and different preparation, but there is the same trend. So thanks to Jason's hard work of thinking of teichoic acid being an antifreeze and thinking of using deuterium NMR, of which we found out there was liquid water there, we've now proven that teichoic acid helps bacteria survive. Okay? And now we have a job where we have to do 1,000 more experiments with lots of different temperatures, lots of different time, lots of different bacteria, lots of different concentrations. Oh, a lot to do. Okay. So the implications are that if you had a soil, mineral, and ice, you could have liquid water around it. And if you look at ice, there's dirt in ice. Okay, hopefully not in this ice, but in regular ice out there. If you go look at, it doesn't. Does it snow up here very much? You guys have been to college. You guys have seen snow, right? And snow gets kind of dirty. Okay, there can be dirt in snow. So you can have soil minerals that will prevent ice formation and provide a small bit of liquid water. The bacterium has teichoic acid, so maybe inside the ice crystal, the bacterium can actually carve out its own liquid water shell and grow around and find dissolved minerals, dissolved sugars, and nutrients with chemotaxis in the ice, which would be interesting to study. But this is one of the possibilities. The other thing, you could have soils and bacteria and teichoic acid to provide a large channel of liquid water. You could even work with other symbiotic species, for instance, gram-negative bacteria or yeast or something else in biofilm communities. This is data for E. coli that we did. That's if you freeze it in PBS without anything added, you get 0 signal. If you freeze it at -20 but add teichoic acid, it rescues and it survives. So now we have to understand why does it survive at about a 10% rate. So this is 1 week. This is not a growth curve. This is just a single data point. So we're beginning to do our studies of showing bacteria are alive when they're frozen. We're beginning to study other species and other microorganisms. We have some mutants. And so what I'd like to do is just finish up by saying, I talked about how we had metal chelation. We have liquid water, which now implication for cryoprotection of microbiology. The fascinating thing about this project is what I didn't realize until maybe a couple of years ago was that usually molecular biology or biochemistry knows the function of an enzyme. They know the function of a protein. And then they go to the NMR jocks and they say, tell us the structure. Why does this enzyme work? Why does this substrate work? Do the X-ray, do the NMR, do something. Tell us why it works. Well, now I get to reverse the tables. I do NMR and I go back to biology and I say, ha ha ha, guess what your molecule does? It's an antifreeze. And it protects life. And you know what you need to do? You need to rewrite that textbook of yours. And you need to add new chapters on teichoic acid. And so it isn't very often that physical chemistry gets to tell microbiology where to go. And so it's the fun thing about science is that when you talk to people, I'm not afraid to ask these questions and do these experiments. So obviously I have to thank Jason for all of his hard work. And I have to thank Amy and Malcolm. Malcolm just last semester spent 6 weeks at NASA on a paid internship at the Jet Propulsion Laboratory. He was working on cleaning spacecraft prior to launch with the Biotechnology Protection Group. So the people at NASA, before they send up the rovers, they don't want to contaminate Mars, they don't want to contaminate the space station, so they have to scrub everything clean and get rid of all the bacteria because you don't want the staph infections on the space station. So what they do is they actually use hydrogen peroxide or UV light to sterilize the spacecraft. So we're working with NASA on other projects of bacterial spores that are resistant to sterilization and cleaning. Here are some postdocs and other professors. Chris Burbas at Northwestern. He necessarily didn't work on this project, but he certainly did a lot of work in the lab and helped us be productive. some current graduate students, and a postdoctoral researcher. I have to thank my colleagues at OU who helped us get started in growing bacteria, Eric Brown, who's a Tacoic acid expert, Todd and Margaret, who have NMR spectrometers, Shahar Mobashiri in Notre Dame, who's done some theoretical calculations. The University of Oklahoma, we have the big grant from NIH, and we have some beginning to work with NASA. But I really, really have to thank Jason, because without this, I wouldn't be here today. And I can't express my gratitude enough to Jason for all of his hard work and dedication. And the dedication that he showed in my lab, I can tell that he's showing here at Northwestern. And I think that's great that you've kept up that hard work. So thank you for your time, and I'd be happy to answer any questions.
In your graph,